Dear student, how are you? I hope you are well. Welcome to my class. I am Samresh Mondol, Senior Lecturer, Department of Biology, Milestone College. Do you know them? First person name is Imbeden, second person name is Mayarov, and third person name is Barnes. These three scientists discovered a pathway of respiration. Do you know about this pathway? This pathway is glycolysis. This glycolysis is related to the chapter 9. From this class, we can know uh, what is glycolysis, what is known as EMP pathway, ATP calculation in glycolysis process. We can describe the glycolysis process. In this process, we have to know about, at first know about what is respiration. RESPIRATION. Respiration is a physiological or biochemical process in which respiratory element break down in presence of oxygen or in absence of oxygen and produce ATP. This process is known as respiration. And respiration are respiration is two type. First one is aerobic respiration. This is basically in presence of oxygen or in absence of oxygen. First one is aerobic respiration. Aerobic respiration. And second one is known as anaerobic respiration. And AER aerobic respiration. Aerobic respiration uh, are respiration are three step. In aerobic respiration there are present three step of aerobic respiration. First one is glycolysis. Glycolysis. First aerobic respiration. First glycolysis. Second Krebs cycle. KREBS Krebs cycle. And third one is ETS, electron transport system, or ETC, electron transport chain. Anaerobic respiration completed by two steps, and first step is glycolysis, and second step is incomplete oxidation of oxidation of pyruvic acid. So we can find out that the common stage of aerobic and anaerobic respiration is glycolysis. In both process, glycolysis is the first step, and this step occurred in cytoplasm, but rest of these two stages occurred in mitochondria, and two steps of anaerobic respiration occurred in cytoplasm. So glycolysis is also known as the cytoplasmic respiration, also known as cytoplasm. Uh, Today's topic is glycolysis. In glycolysis process, uh, at first glucose breakdown step by step in presence of different enzyme and at last produce two molecules pyruvic acid. So at first let me write down glucose. Glucose. We know that glucose is a 6 carbon base monosaccharide and aldehyde group based. And symbol of glucose is C6H12O6. At first glucose in this process produce glucose 6 phosphate. Glucose 6 phosphate. Glucose at first produce glucose 6 phosphate. Difference between glucose and glucose 6 phosphate is 1 phosphate which is remain at the carbon number 6, and this phosphate supplied by ATP. ATP means adenosine triphosphate. This adenosine triphosphate gives 1 phosphate and turns into ADP, adenosine diphosphate. When ATP turns into ATP or ADP turns into ATP, then they are needed kinase enzyme. Basic enzyme name kinase. This kinase enzyme works on glucose. For that reason, this kinase enzyme main name, total name is exokinase. That means glucose 6 carbon. In glucose they are present 6 carbon. So enzyme name exokinase, and this reaction occurred in presence of magnesium ion. Next, this reaction is a non-reversible reaction. In second reaction, glucose 6 phosphate turns into fructose 6 phosphate. Fructose 6 phosphate. Difference between glucose and fructose is in glucose they are present aldehyde group and in fructose they are present ketone group, and both are isomer to each other. So they are needed isomerase enzyme, and this reaction is a reversible reaction. An enzyme name phosphoglucoisomerase. That means at first we have to write down the name of side chain, name, then main body of substrate, and next name the main enzyme name. So enzyme name phosphoglucoisomerase. Phosphoglucoisomerase. Next, fructose 6 phosphate turns into fructose 1,6 bisphosphate. Fructose fructose on 6 bisphosphate. So this reaction is a second reaction. When fructose 6 phosphate turns into fructose on 6 bisphosphate, what's the difference? 1 phosphate, and this phosphate supplied by ATP. ATP then turns into ADP, and this is a non-reversible reaction. So enzyme name will be kinase, phosphofructokinase. Phosphofructokinase. kinase. Next, fructose on 6 bisphosphate breakdown and produce two compound. Produce two compound. First one is 3-phosphoglyceraldehyde. 3-phosphoglyceraldehyde (COD) and dihydroxyacetone phosphate. Dihydroxyacetone phosphate. This reaction is a reversible reaction and enzyme name aldolase. Enzyme name aldolase. Aldolase. That means bis-phosphofructoaldolase. Side chain, main body and main enzyme name bis-phosphofructoaldolase. We can also write down the name of this aldolase enzyme bis-phosphofructoaldolase. Next, this 3-phosphoglyceraldehyde turns into on 3-bisphosphoglyceric acid. On 3-bisphosphoglyceric acid. The difference between that means aldehyde turns into acid. When aldehyde turns into acid, then NAD turns into NADH plus. Also, 3-phospho turns into on 3-bisphospho. That means they are needed a phosphate. This phosphate added as a inorganic phosphate. They are also present water. As in this reaction, hydrogen release, they are needed dehydrogenase enzyme. That means phosphoglycerol dehydrogenase. Phospho enzyme name will be phosphoglycerol dehyde, phosphoglycerol dehyde, phosphoglycerol dehyde dehydrogenase. Dehydrogenase. Next reaction, 3-phosphoglyceraldehyde then turns into 1,3-bisphosphoglyceraldehyde then turns into 3-phosphoglyceric. This reaction is also a reversible reaction. When 1,3-bisphosphoglyceric acid then turns into 3-phosphoglyceric acid. The difference between 1,3-bisphosphoglyceric acid and 3-phosphoglyceric acid is a phosphate. That means on phosphate release, and this release phosphate received by ADP, adenosine diphosphate, and turns into ATP, adenosine triphosphate. So basic enzyme name kinase and full name phosphoglyceric acid kinase. phosphoglyceric acid kinase. Phosphoglyceric acid, glyceric acid kinase. This is also a reversible reaction. Next, 3-phosphoglyceric acid then turns into 2-phosphoglyceric acid. This is also a reversible reaction. 2 phospho phospho 2-phosphoglyceric acid. The difference between 3-phosphoglyceric acid and 2-phosphoglyceric acid. In 3-phosphoglyceric acid, phosphate remain at carbon number 3, and 2-phosphoglyceric acid, this phosphate remain in carbon number 2. So there occurred a rearrangement of side chain. This process is known as a mutation and enzyme name mutase. Complete name phosphoglyceric acid mutase. Phosphoglyceric acid mutase. Next, 2-phosphoglyceric acid then turns into 2-phosphoenol pyruvic acid. 2-phosphoenol 2-phosphoenol pyruvic acid. Pyruvic acid. This is a inhalation process, and this process occurred in presence of this process. When this process occurred, then they are released on molecule water, and enzyme name enolase enzyme. That means phosphoglyceric acid enolase, or we can easily write down only enolase enzyme. This is also a reversible reaction. Next, at last 2-phosphoenol pyruvic acid at last turns into pyruvic acid. Pyruvic acid. When 2-phosphoenol pyruvic acid turns into pyruvic acid, then one molecule inorganic phosphate release this phosphate received by ADP, and then ADP turns into ATP. Enzyme name kinase, main enzyme name, and complete name phosphoenol pyruvic acid or pyruvate kinase. Pyruvate kinase or phosphoenol pyruvic acid kinase. This pyruvic acid is a 3-carbon base compound CH3 CO COOH. In this pyruvic acid there are present 3 carbon, but glucose is a 6-carbon compound. In this case, then dihydroxyxis, when fructose 1,6-bisphosphate breakdown produce 3-phosphoglyceraldehyde and dihydroxyacetone phosphate. In this time, dihydroxyacetone phosphate turns into 3-phosphoglyceraldehyde in presence of isomerase enzyme. They are isomer to each other and produce again 3-phosphoglyceraldehyde. And this side reaction from 3-phosphoglyceraldehyde to pyruvic acid, these reactions occur twice or two times. That means if we number the reaction, this one is the third reaction, fourth reaction, fifth, 6, 7, 8, 9. This 5 to 9 nong reaction occur twice or 2 times. So in glycolysis process, they are produced two molecules NADH plus, two molecules in reaction number 5, two molecules water release. In reaction number 6, they are produced two molecules ATP. So 5 to 9 nong reaction, this reaction occurred twice or 2 times. If we want to describe this every reaction, we can easily describe this reaction. We have to write down at first the name of the enzyme name. If we describe the on-nong reaction, at first we have to write down the enzyme name. Enzyme name hexokinase. Next, we have to write down the substrate. Substrate name glucose. We have to next write down the product. Product name glucose 6-phosphate. At last, we have to write down the name of byproduct. That means ADP turns into ADP. In presence of total description will be in presence of hexokinase enzyme and magnesium ion, glucose turns into glucose 6-phosphate. In this case, ATP turns into ADP. Reaction number 2, in presence of phosphoglucoisomerase, glucose turns into fructose 6-phosphate. In reaction number 3, phosphofructokinase, in presence of phosphofructokinase and magnesium ion, fructose 6-phosphate turns into fructose 1,6-bisphosphate. By this way, we can easily describe all the reaction. This glycolysis process occurred in cytoplasm. So this process also known as cytoplasmic respiration. And this step invented by three scientists named Embeden, Meyerhoff and Parnes. For that reason, this step also known as EMP pathway. In this glycolysis process, we can find out that at first they are used 2 ATP, and in reaction nung 5 and in reaction nung in reaction nung 6 and reaction nung 9, they are produced 4 ATP. 6 and 9 nung reaction, they are produced 4 ATP. So they remain 2 ATP. In this process also produce 2 NADH plus. This NADH plus break down in ETS process, and they are from 1 NADH plus they are produced 3 ATP. So for 2 NADH plus they are produced 6 ATP. So from 2 NADH2 they are produced 6 ATP. Total ATP production will be in glycolysis process 6 to 8. In glycolysis process, total ATP produce 8 molecules. By this way, we can easily calculate the number of ATP in glycolysis process. I hope you can easily understand the glycolysis process. Now we can write this glycolysis process. If you fail to understand any reaction or this process or any place, please contact your subject teacher. Hope you will comfortable